morning, friends. Good morning, students. In today's class, we will discuss pyrimidine nucleotide synthesis and breakdown, right? So, I will share mnemonics as well to remember the pathway and the enzymes. So, do watch the video till the end, right? Okay. So, first of all, how to remember the name of pyrimidines? CUT, cut. C for cytosine, right? Then U for uracil and T for thymine, right? So to convert them into their corresponding nucleotide, use CMP, UMP, and TMP. So cytidine monophosphate, uridine monophosphate, and thymidine monophosphate, right? Now coming on to this molecule, pyrimidine, right? So remember the first thing that the numbering is clockwise, right? So I will number them 1, 2, 3, 4, 5, and 6, right? So now the individual components came from, so I will distinguish them from which part, the which where it is derived, right? So half of the molecule, this one is derived from aspartate, right? And this second carbon is derived from carbon dioxide, right? And the third nitrogen is derived from glutamine, right? Numbering is clockwise, right? And the half of the molecule is derived from aspartate, second carbon is derived from carbon dioxide, and N3 is derived from glutamate, right? Now I'll start with the synthesis and I will share mnemonic. Now to remember the steps, I had made a mnemonic, that is, cool, creative people can do outstanding operations and manage people by using mind power, right? So starting is by cool creative people, right? Cool for carbon dioxide, right? So carbon dioxide is the basic requirement to start the pyrimidine biosynthesis, right? So carbon dioxide will combine in glutamine, right? And here we'll use two ATP molecules, right? And we'll get two ADP, right? And this reaction is catalyzed by CPS2, right? And this is a cytosolic enzyme, and the CPS1 is used in urea cycle synthesis and CPS2 is used here, right? And it is a cytosolic enzyme. In that case, it was mitochondrial enzyme, right? Then the second step, can cool creating people. For creating people, the second mnemonic is carbamyl phosphate, right? And the second enzyme is ATC, aspartate transcarbamylase, right? And we'll add aspartate here, right? And we'll get phosphate, right? And in the third step, creative people and can, Ca and can, Ca for carbamyl aspartate, right? And our third product is formed, that is our carbamyl aspartate. And after that, carbamyl aspartate, do for dihydroorotate, right? And in the third step, we'll remove a molecule of water, and the enzyme is for D is dihydroorotate, right? In the fourth step, we'll get dihydroorotate, right? And from dihydroorotate, we'll get our next product, that is our orotate, right? And orotate is formed by DH, our next enzyme, that is dehydrogenase reaction. And here we use NAD, and this converted to NADH plus H positive, right? Right. Fourth step is clear. And dihydroorotate, and we'll get orotate. From orotate, we'll get our OMP, that is our orotidine monophosphate. And from orotate, we'll get our OMP, and this reaction is catalyzed by PRPP, phosphoribosyl pyrophosphate dependent reaction. And here the enzyme is orotidine phosphoribosyl transferase, right? From here we'll get our OMP, orotidine monophosphate, and it is converted to uridine monophosphate. And in this step we and from OMP there is a decarboxylation reaction, and carbon dioxide is removed, and the enzyme is OMP decarboxylase, right? And we'll get our final product, that is our UMP. From UMP we can form UDP, right, by kinase reaction, right? And from UDP we can form UTP triphosphate by kinase reaction, right? From GTP we can form CTP cytidine triphosphate by our reaction by donating ATP and glutamine, right? And this UDP can be converted to deoxy UDP by ribonucleotide reductase reaction. From deoxy UDP you can form deoxy UMP, and deoxy UMP can go to TMP, thymidine monophosphate by thymidylate synthetase reaction, right? Get TMP. So finally, to revise, cool for carbon dioxide, creative people for carbamide phosphate, can for Ca for carbamide aspartate, then do for dihydroorotate, then outstanding for orotate, right? Operations and manage people OMP, right? And by using mind power for UMP. From UMP, we'll get diphosphate by kinase reaction. From di, we'll get again triphosphate by kinase reaction. From UTP by ATP reaction and glutamine, we'll get our CTP. And from UDP by ribonucleotide reductase reaction, we'll get deoxy UDP. From deoxy UDP, we'll get UMP. And by thymidylate synthesis reaction, we get our thymidine monophosphate. So we synthesize all the pyrimidines and the enzymes by the sequence CAD, C for carbon phosphate synthetase 2, A for aspartate transcarbamylase, D for dihydroorotase, and DH for dehydrogenase, O for ornithine phosphoriboside transferase, and O for ornithine monophosphate decarboxylase. So the sequence has well, and now I will start with degradation. Now to start with degradation, we have UDP, CTP, and thymidine triphosphate, right? These triphosphates are corresponding to their corresponding uracil, cytosine, and thymine, right? So we have removed the phosphates by dephosphorylase reaction and amino groups by deaminase reaction, and finally we're getting uracil and cytosine and thymine, right? For further breakdown, uracil and thymine, they are converted to dihydrouracil, then to form beta alanine, and beta alanine is converted to acetyl CoA, and acetyl CoA can enter TCA cycle and further metabolize, right? And in case of thymidine triphosphate, it undergoes dephosphorylation reaction, deamination reaction, and glycosidic bonds they break down, and we'll get our thymine. Thymine will be converted into dihydrothymine and then to beta minus isobutyrate. From here it is converted to succinyl CoA. From succinyl CoA it enters the TCA cycle and further metabolized. Right? So that finishes with our degradation. Now let's start with regulation. Start with the regulation of pyrimidine synthesis. It is regulated two levels. First is the enzyme aspartate transcarbamylase, and second is carbamyl phosphate synthetase 2, right? And ATC is a completed step in the regulation, right? And it is regulated by end product, right? Feedback mechanism of end product, right? PRPP activates it and UTP inhibits it, right? And in case of CPS2, phosphoribosyl pyrophosphate ATP, they activate it, and UTP and UDP, they inhibit it, right? It is by end product inhibition, it is by it is by feedback inhibition, and it is by the end product feedback regulation of end product, right? So that finishes with our chapter on pyrimidine synthesis, breakdown, and regulation. For upcoming videos, please like the channel and subscribe it and press the bell button below. Thank you. Keep sharing, keep studying. God bless.